Hi, everybody. So we've been talking about how different taxa acquire and process nutrients. And just to recap where we've been, we've talked about the nutrients that all organisms need. We've talked about some generalities of nutrient acquisition and different types of trophy. So photo, auto, chemo, hetero, that sort of thing. We've talked about themes for acquiring and transporting nutrients, surface area to volume ratios, and some of the general mechanisms that organisms used to get nutrients into their individual cells. And so now what we're going to talk about is how plants acquire and transport nutrients. And just as a reminder, if you haven't already watched the lectures that cover these general topics, please make sure you view those before you move on into this lecture. So just as a quick reminder, we're going to be talking about adaptations that organisms use to obtain and transport nutrients. So obtain nutrients from the environment and transport them within their sort of individual bodies or cells. And the main examples we'll be talking about are photoautotrophs and chemoheterotrophs. So just as a quick reminder, photoautotrophy and chemoheterotrophy is all about how organisms acquire energy and carbon. So all organisms can be broken down into either phototrophs or chemotrophs. Phototrophs get energy from sunlight, chemotrophs get energy from chemical compounds, and then they can be further broken down. Some phototrophs are autotrophic, so they get carbon sources, to make carbon-to-carbon biological molecules, either from inorganic sources like CO2, which is what we see in plants, or they can get them from organic compounds, so eating other organisms, and that's heterotrophy. So animals are an example of, for example, chemoheterotrophs, and plants are an example of photoautotrophs. And so we're going to be talking about plants in general as our representative taxa for photoautotrophy, and we're going to talk about fungi and animals as our representative taxa for chemoheterotrophy. Don't forget that there also are chemoautotrophs and photoheterotrophs, but it's only microbes that can do photoheterotrophy and chemoautotrophy. And there's some examples that are here, but please just review your old lectures about prokaryote metabolisms to review chemoautotrophy and photoheterotrophy. We're not going to go into those in great detail here in these sets of lectures. So let's begin talking about our representative photoautotroph, so the vascular plants. So just a quick review of plants. Remember, plants are eukaryotes. They're this sort of kingdom that are down here. And within the plants, there's this group called the green plants, and within the green plants, there's this group called the land plants. But all of the plants in general, they are a monophyletic group. They are all eukaryotes. They are all photosynthetic, and they all have cell walls. Okay, so if we were to zoom in on this group here, looking at the green plants and the land plant group, you can break these groups further down. Remember, we're going to be talking about the vascular plants. So if you were to decide where vascular tissue goes on this tree, where would you put it? All right, so if you said point D, you are correct. So these are the vascular plants. They have vascular tissue. And recall that vascular tissue are basically the xylem and the phloem, these groups of sort of tissues and straws that form these tubes that run through the body of a plant to transport sugar and water and various minerals and nutrients like phosphorus, potassium, nitrogen, and other key elements that play a role in forming the macromolecules that plants and all organisms need to survive. And so most of this stuff should be a review for you guys at this point. So let's break this down a little bit further and talk about how plants acquire and transport nutrients. In general, there are two general body systems, body in quotation marks because you don't normally refer to a plant's body, but essentially you've got an above ground shoot system that you're looking at here that's composed of the branches and leaves and trunk of the plant. And the main job of that above ground shoot system is to collect CO2 and light. And it's collecting CO2 and light so that that shoot system can produce sugars and other important carbon-to-carbon molecules, so biological molecules that are carbon-based molecules, through the synthesis of carbon-to-carbon bonding. And photosynthesis actually forms these carbon-to-carbon molecules. So it links the carbon molecules in CO2 together into more energy-rich, reduced carbon-to-carbon molecules like sugars. And then it'll transport those sugars and other carbon-to-carbon molecules to the rest of the plant. And the rest of the plant is the root system. Okay, so this is the below ground system of roots, and the main job of the root system is to collect water and minerals and other nutrients and transport those water and mineral nutrients to the rest of the plant. So let's first look at this shoot system in a little bit more detail and the role it plays in collecting CO2 and light and producing some of the key nutrients that plants need. So remember, typically leaves in the shoot system collect CO2 and light for photosynthesis. And this equation represents photosynthesis, where you have CO2 and H2O reacting with one another to form glucose, this sort of complex reduced high energy molecule, and O2 as a byproduct. And so these carbon molecules, these CO2 molecules that are collected from the air, are being reduced into this higher energy and transferred into this higher energy sugar molecule. And so the job of leaves is to be able to collect this CO2 from the air, and also light. So light is a sort of the energy that goes into this reaction to reduce this CO2 molecule, and that light energy is converted into the bonds of this sugar molecule. So you have light energy being converted into chemical energy. And so leaves need to collect CO2 and light for this reaction. And their surface area to volume ratio is really important for this light collection and CO2 collection. So their structure is related to their function. And having a really high surface area to volume ratio to exchange gases and collect light for the environment is really important to just enhance light collection, enhance CO2 collection, and enhance sugar production. And so having a really big, large, flat leaf with a large surface area to volume ratio, and also having high densities of stomata for more efficient gas exchange. So remember, stomata are these little holes in the bottoms of leaves where CO2 can get in. So the more of these holes that you can have to collect CO2, once again, the more photosynthesis you can do. The larger surface area to volume ratio the large flat leaves have will maximize light and CO2 collection, maximize photosynthesis, maximize sugar production, maximize sort of nutrient intake for these plants. But remember, there's a trade-off here. So often CO2 uptake is a trade-off with water regulation. So the more CO2 that gets in from these stomata, the more water that can get out through the stomata. And so plants that are in water-limited environments or drier environments tend to want to reduce the water loss at the cost of lower sugar production. And so they'll tend to have smaller leaves, actually reduce the surface area to volume ratio so that they're not losing as much water. They'll tend to have fewer stomata, and they'll tend to time the opening and closing of stomata so that they don't lose as much water. And so, for example, you can see this cactus here with all these different thorns on it or spines on it. Those spines are actually reduced leaves. They've been reduced so small that they're not even leaves anymore. They're now thorns, and it's actually the trunk of the cactus that's photosynthetic. And once again, this is all about water conservation, minimizing water loss at the cost of nutrient production. So this whole idea of structure and function and leaf adaptations to collect CO2 and light, but also minimize water loss. One thing that they can do is regulate the opening and closing of stomata. Remember, CO2 is getting in through the stomata, water is leaving through the stomata. So typically at wetter and cooler times of either the year or the day, the stomata tend to be open. And at hotter and drier times of the year or day, the stomata tend to be closed. The other thing is you can see variation in stomatal densities within different plants and also within different species, such that in some plants you'll have much higher densities of stomata, and in other plants you'll have much lower densities of stomata. So which would you predict in a drier environment, A or B? All right, so if you said B, you are correct. This is what you typically see in a drier environment, lower stomatal densities. And that's simply because, you know, light is not a limiting factor. Water is the limiting factor here. And so they want to limit water loss or reduce water loss. Whereas in a wetter environment, the water is not a limiting factor. You want to actually increase CO2 gain as much as possible. And it doesn't really matter if you're losing a lot of water. So the more holes you can have to sort of collect CO2 and increase photosynthesis, the better. Other leaf adaptations to maximize CO2 and light collection and also minimize water loss is typically in wetter, lower light conditions. Plants will have a much higher surface area to volume ratio, like you see in these giant leaves here. Here they're just trying to maximize light collection, and it doesn't really matter if they're losing a lot of water because water is not a limiting factor. This is a more tropical plant in a tropical environment. Whereas in drier, higher light conditions, plants tend to have lower surface area to volume ratios, and we tend to see convergent evolution. So remember, this is different species independently evolving for the same sort of phenotypes, where you have shrunken leaves, spines, succulents. So if you think of succulent plants, they have very thickened, waxy cuticle. And they also tend to have more, will have photosynthetic stems. And these are all adaptations to reduce water loss. And we see this in 3 different families of plants. We see this in the euphorbs, in the cactus plants, which are a different family. So remember, family is a taxonomic group. And also in other succulent plants, we see convergent evolution for these sort of phenotypes that reduce water loss. So all of these plants are from different families, and they've convergently evolved to have this sort of cactus look to them. The last thing I want to talk about is how this shoot system transports sugars that are produced in the leaves to the rest of the plant through this process of bulk flow. So remember, we talked about bulk flow in multicellular organisms. Bulk flow is really important because although diffusion can move individual molecules from places of higher concentration to lower concentration, the rates of diffusion are usually not fast enough and sufficient to get important solutes, important nutrients to all parts of a large multicellular organism and all the cells of a multicellular organism. So multicellular organisms typically rely on bulk flow to transport nutrients over large distances and get them to every single sort of cell. And so phloem is one type of vascular tissue that's really important for this bulk flow in plants because it transports carbohydrates from sources to the rest of the plant body and from sources to sinks. So sources are places that produce lots of sugars and carbohydrates, and sinks are places that don't produce them and require them. So let's look, for example, at this breakdown of a plant, and here we're looking at a zoom in of the leaf, and we're looking at a zoom in down below of the roots. And what you can see here is you're looking at the xylem, where water's flowing from the roots up to the rest of the plant, and then you're looking at the phloem here, where you have carbohydrate flow. And so in the source cells, here we're looking at a leaf cell, and in that leaf cell, you've got a cell wall, you've got a cell membrane, you've got a vacuole, you've got mitochondria, and then you've got these chloroplasts where photosynthesis is happening. So if photosynthesis is happening, these chloroplasts are producing a lot of sugars that get converted into a particular type of sugar called sucrose. And so all these little dots here represent high concentrations of sucrose. These are sugars that are being produced through photosynthesis. And what happens is those sucrose molecules get highly concentrated in this companion cell, and then they'll move by diffusion at a sort of low rate into the phloem. And what happens is essentially they'll move from these high concentrations to lower concentration cells in the roots. And so this is considered a source, this is considered a sink. And the way it moves is through essentially what's called an osmotic pump. And so basically the plant regulates concentrations so that water through osmosis is flowing from source areas to sink areas following the sort of higher to lower concentrations of sugars, and that will cause the water to flow from sources to sinks. That water will bring the sugars down through the phloem into the sink cells. And so here you can see a root cell. That root cell also has mitochondria. It may also have some chloroplasts in it, but those chloroplasts, because they're not exposed to light, aren't producing as many sugars. And so in order for these sink cells to survive, they need sugars. And so those sugars get shipped from the leaf cells that are doing photosynthesis as a source, move down the phloem to the sink cells that need those sugars. And so why is sugar needed in these sink cells? Go ahead and answer that question. Why do these sink cells need sugar? So other cells need sugars to do cellular respiration and make ATP. So you'll notice that all these plant cells, they have mitochondria. The sink cells have mitochondria. All plant cells, they need to have mitochondria. Eukaryotic cells have mitochondria. And those mitochondria take those sugars. They do cellular respiration to turn those sugars into ATP, because all eukaryotic cells need that cell, and pretty much all living cells need ATP because that's a currency that cells use to do a lot of other cellular level reactions. Okay, which of the following statements is correct? All right, if you said plant shoots collect CO2 and light to make sugars while roots collect nutrients and water, that is correct. So let's look at this first one. Why is this one wrong? Sugars flow through the xylem from the roots to the leaves. Okay, so first of all, sugars flow through the phloem, not the xylem, and they go from the leaves, which are sources, to the roots, which are sinks. So this is wrong for several reasons. Let's look at B. Leaves often have a low surface area to volume ratio to increase absorption of water and light. This is false. Leaves often have a high surface area to volume ratio to increase the absorption of CO2 and light, unless they're in a dry environment. Plants in drier environments typically have leaves with high stomatal densities. This is false. They typically have lower stomatal densities, and that's to reduce water loss. And they typically have lower surface area to volume ratios, also to reduce water loss. And gymnosperms, angiosperms, mosses, and ferns have vascular tissues. That's false. Mosses are non-vascular plants. They do not have vascular tissues. And this says that the phloem transports water and minerals. This is false. It's the xylem that typically transports water and minerals, not the phloem, which is involved in transporting sugars. Okay, so just to recap, we've talked about all these things up to this point. Today we talked about how plants that are sort of representative of the photoautotrophs acquire and transport nutrients. We talked about the shoot system for generating sugar and generating carbon-to-carbon molecules, so these biological molecules, and the root system playing a role for the transport of water and minerals. We've talked about some of the trade-offs in the structure and function of leaves and their role in collecting light and CO2, but also doing this without losing too much H2O. And we talked about how leaf size and shape and stomatal and densities can influence the rates of light collection, CO2 collection, and also water loss. And then lastly, we talked about the role of phloem in using bulk flow to transport sugars from sources to sinks. So high concentrations of sugar to lower concentrations of sugar, typically from the leaves to the roots. And so next, we'll talk about how plants acquire and transport and use nutrients. So we'll continue on this in the next lecture.